Okay. Yeah, I haven't done that before.
Oh, it says recording now. Are you doing that?
Oh,
Okay.
yeah, I just clicked the button. So I'll just...
Okay, I think we should start the webinar. It's a great pleasure to introduce Dr. Mark Zeri. He's a senior scientist of environment and public health at the Institute of Occupational Medicine and also a research fellow at the University of Edinburgh. Mark is kindly already gave us a seminar in the past July, if I'm not mistaken, right, Mark? And the first part of what he's going to present today, a very interesting seminar on the association between area level environmental predictors and all-cause mortality in Scotland, because this is a very relevant topic also for Cyprus. And I believe this kicks very nicely the webinar series of our institute, because we're going to have also another set of webinars for the fall semester here at CII. And we're very happy that you are giving the second webinar today. Mark, the floor is yours.
Excellent. Okay, thanks very much.
Thank you.
I'll share my screen and we'll get started. Okay, great. Yeah, thanks. It's great to be here with you again for the second part of the seminars on spatial epidemiology. So going to talk about a specific example of environmental predictors of COVID-19 deaths in Scotland. So a slight change from what I proposed earlier. Just to recap on last time, we talked about what the constituents of spatial epidemiology are, so disease mapping, spatial clustering, and geographic correlation studies. We focused on spatial clustering last time, and I gave some historical examples. We also talked about how we define degrees of clustering using density-based measures. And we talked about point and areal data. In particular, we looked at how to define spatial autocorrelation using areal data. And that will be really important for today. So yeah, we'll do a recap later on as well about that. And I did an example using R of looking at clustering in aggregated death data. So today we're going to be concentrating on geographic correlation studies. I'll give a couple of examples from the literature, one on mortality from coronary heart disease in England and Wales, and another on poverty and death in inner London. I'll talk about some of the limitations of these types of studies. The main one, which you've probably heard of, is the ecological fallacy. And then we'll talk about spatial dependency. So that's following on from what we did last time. And then I'll talk about some analytical strategies that can be used to reduce these biases, instrumental variables, hybrid models, and spatial models. Well then, so we're getting on, should be around half an hour to that point. We can take a break if people are wanting one. If not, we'll go on to looking at the example in R. So this is using a spatial model to resolve the spatial dependency issue. And we'll be using data on the Scottish COVID-19 deaths. So geographical correlation studies are one part of what are defined as ecological studies. Ecological means that we're interested in groups of people rather than individuals. So it could be at a community level or a country level. So geographical correlation studies are one type of ecological studies. You can also get time trends, also investigations of groups of migrants compared to natives. But today we're interested in geographical correlation studies, and we can use a number of variables. So we can look at aggregate data. So we can these are variables that can be measured at an individual level. So, for example, we can ask people how many cars they own, what their occupation is, and that can give us a sense of their level of deprivation. We can measure that at the individual level, and then we can aggregate those people and get the average for an area. And that's what we call an aggregate variable. We can also get environmental variables. So, the percentage of green space within an area or the level of air pollution, and we can assign that to an individual. Finally, we can get global variables where we can't measure those at an individual level. So these are only applicable to areas. So population density is an example of that, where it doesn't make sense to have an individual being assigned a population density. And so you can see some of the advantages of an ecological design in that we can actually start to look at variables that we can't look at in individual study design. So you might have seen a pyramid like this. It's showing the hierarchy of evidence. It's quite common in epidemiology textbooks, and it shows the weight of evidence, going from the randomized control trial at the top to the case reports at the bottom. And you can see that ecological studies are quite low down there. And so you might ask yourself, well, why do an ecological study if the evidence isn't going to provide, it's not going to be strong evidence. And so what I'd say to that is actually, I kind of disagree with those types of pyramids, because in itself, study design isn't is a insufficient surrogate for risk of bias. You have to also think about lots of different parts of the design that will go into the quality of the study. And so this kind of relates to some of the work recently on defining a new evidence pyramid and trying to appreciate that the quality of the study can make a big difference. With that being said, there's lots of questions that can only be answered with an ecological design. So it might be unethical to randomize a certain exposure to people. It might be unfeasible to measure an exposure in a cohort study. And so ecological designs can be really important for those types of questions. Ecological designs are also really important for stimulating new hypotheses and really are at the cutting edge of epidemiology for that reason. If I haven't convinced you of their utility, I'm going to talk about a couple of examples which really have changed the research agenda in the last 20, 30 years. First one is mortality from coronary heart disease in England and Wales, and this was evidence that was provided for the developmental origins hypothesis. It's also called the fetal origins hypothesis, or the Barker hypothesis, after the lead author of this work. And you can see on the right-hand side there the the graph showing neonatal mortality in 1911, and then 50 years later, the coronary heart disease deaths. You can see that there's a moderate correlation there, and it's showing, it's providing evidence that those early life exposures are important for your health trajectory across the whole of your lifetime. And why is, so the kind of initial evidence for this hypothesis was from geographical correlation studies, and that's produced a huge impact on what we've the type of studies that we've seen thereafter. When the reasons for these different trajectories are ascribed to the individual life circumstances, we term that body of work life course epidemiology, which is a huge part of epidemiology now. When we look at another example of poverty and death in England, you can see from the right-hand side the levels of poverty in London at the top from 1896, and then in 1991 being remarkably similar. You can see that you can see visually the correlation with death. So you can see that the spatial patterns of deprivation and mortality have been robust for over 100 years. And what these correlation studies have done have stimulated a look at place-based variables and how they've changed over time, and what that has meant for health in later life. And this is kind of stimulating a body of work called the life course of place. So myself and co-authors have looked into this work, and have looked at how Edinburgh has changed over the last 100 years, and looked at how different parts of different aspects of places have changed the risk of cognitive decline in later life. So some people will still not be happy with an ecological design, and that's really because of a few different limitations. The one that you might have heard of already is the ecological fallacy. So what that means is that we cannot, well, it means that the relationships that we see at an area level don't play out at an individual level. And the two main sources of that are when we've omitted confounders, and when there is also omitted effect modifiers. So the classic example is where we have Robinson, who studied the 1930 US Census, and he found that at the state level, the percentage Black ethnicity was strongly correlated with the percentage illiterate. But he didn't see that at the individual level. So when comparing Black against other and illiteracy, it was a weak correlation. So this is often cited as a historical example of the ecological bias, but it's kind of still the reason why people are wary of ecological studies and also geographical correlation studies. The second big limitation is one that we've discussed previously. So it's around the concept of spatial autocorrelation. Just a reminder, spatial autocorrelation is where units that are in close proximity to each other are more similar. And it can be a problem with when it's not accounted for in regression models. So it can result in incorrect conclusions because you don't have independence of residuals. So what can we do to strengthen ecological studies, to make them, to provide stronger evidence and to resolve the limitations that we talked about? There's a few different options. We can apply instrumental variables. We can look at hybrid models, so two-phase design where we look at ecological and individual study designs. We can look at multi-level models, and then we can also, so those two are addressing the the ecological bias. The third one is looking at resolving the spatial dependency problem. And yeah, so we can apply spatial models to account for autocorrelation. And the two main ones are SAR and CAR, and we'll talk about those in a couple of slides. So first of all, instrumental variables. So some people have argued that ecological analyses are only useful when and meaningful when the ecological variable can be interpreted as an instrumental variable. So what is an instrumental variable? If you look on the right-hand side, you'll see the directed acyclic graphs showing the relationships between the different variables. So the instrument is associated with the exposure of interest. The instrument is associated with the outcome only through the exposure of interest. And finally, the instrument is not associated with the confounding factors. So that's the three criteria that you need to have a sensible instrument for your study. And if we go on to the second row, we can see that this is an example of Mendelian randomization. So we've got we can have a genotype which encodes for a certain phenotype and is then related to health outcome. So in my line of work, we considering UV exposure and vitamin D levels. We know that there are single nucleotide polymorphisms that encode for the vitamin D binding protein. So that will allow circulating vitamin D to be transported all around the body. And it means that the people that have kind of disruption to this SNP have lower levels of vitamin D. And then we can look causally at what that means for their health outcomes. So just the final row, I just wanted to show an example of how we can do it in an ecological setting. So this is one that I've done recently with co-authors, looking at the effect of ambient UVA, so ultraviolet radiation A, on COVID-19 death. And we're able to do that because at the area level, ambient UVA is associated with that individual UVA exposure. And we're proposing that UVA exposure is related to COVID-19 death through increased production of nitric oxide, which reduces blood pressure. And as you know, blood pressure, hypertension is associated with higher risk of death. So that's an example of how we can use it in an ecological setting. Another way to resolve ecological bias is to repeat the study with individual data. So this is a two-phase design. So we look at the results from an ecological study, we compare them to an individual level study, and if we see a similarity in the effects, we can rule out that the ecological bias is a problem. So some of the work that has done this is on air pollution. Duncan Lee and Christoph Saaren have shown that the models that they've built using air pollution in an ecological setting can be replicated in individual level models. Finally, for looking at ecological bias, we can do a multi-level study design. So before we were designing separate individual and ecological studies, multi-level studies are ones that bring them both together. So in the case of socioeconomic status and health, we can understand the effect of living in a deprived area, but also the effect of being low income in that deprived area. And what we do is we, to apply multi-level models, we augment the standard regression, which has your covariates, which are thought of as fixed effects, and then you bring in random effects, which you can think of as grouping factors. So they're taking account the hierarchical structure of your data. So in the example above, we've got an individual and they're nested within an area. And so by including random effects, we're accounting for the correlation that you would expect in responses for people that live in the same area. And importantly, they can be thought of as a random, the groups are a random sample of the much wider number of groups. And so you can infer out with the groups that you've got in your model. So that's a big advantage of doing a multi-level model rather than, what you could do is just put in dummy variables for all your groups. But what that wouldn't allow you to do is infer outside of the groups that are in your model. Also, what it would not allow you to do is look at the difference between group effects and group predictors, because in that kind of dummy variable model, they would be completely correlated with one another. So one of the kind of key outputs from a multi-level model that give you a good understanding of what the effect of the grouping structure is it's called the intraclass correlation coefficient, and what that allows you to do is look at the percentage of variance that's explained by your grouping factors. So there's a number of software that you can use. The one that provides you with the GUI is MLWin from Bristol University. In R, the most common package is LME4. It stands for Linear Mixed Effects 4. And that's one of the slight issues with multi-level models, is that they're called different things to different people. So some people will call them mixed effects models, some people will call them multi-level models. They're also called random coefficient models. I think another one is variance component model. So yeah, it's worth remembering that, I guess, if you're looking for software and also help online. Okay, so the other thing that we want to resolve to strengthen our ecological study is to account for spatial dependency, and we can do that with simultaneous autoregressive random effects. So just like we put in a random effect to account for the kind of nested structure in our data set, we're now going to put in a random effect to incorporate spatial autocorrelation. So the kind of the fact that neighboring units will be more alike. And so SAR models are used quite commonly, and you'll see that they're defined into lag models and error models. The difference between those is how you define the kind of correlation. The lag model is defining your correlation between the dependent variable, whereas the error is defining the dependence between the errors. So a great place to start with these types of models to see whether it makes a difference to your normal linear regression model is 𝗔𝘀𝘀. And if you want to replicate these models in R, the spatial reg package is the one to go for. There are also another set of models called conditional autoregressive models. So these are similar, but slightly different in the way that they operationalize that covariance structure. Pragmatically, what it means is that CAR is more appropriate for situations where you've got first order dependency, and SAR is more appropriate when you've got second order. So if we think back to last time, I showed you the adjacency for, so you can see the diagram on the bottom right there. On the left-hand side, you can see the 𝗃 adjacency, and on the right-hand side, you can see the Queen adjacency. And so what CAR is appropriate for is when you believe the dependence is like the green. It's just the neighbors, the immediate neighbors. Whereas if you think that the dependency is more like, includes the yellow, then a SAR model is more appropriate. So you can fit these models in a frequentist paradigm. So some of the software there is the SPAM model, so the spatial mixed model, and HDLM. I definitely recommend if you've got larger data sets to use HDLM. It's much more efficient. For an example, the data that we're looking at today later on has around 1,000 units, observation units. It takes about a minute in SPAM. So I'd say anything over 10,000 units, you'd probably be better to use HDLM. You can also fit these models in a Bayesian paradigm, Bayesian approach. So you're adding that prior information in to get posterior distributions for the CAR random effects, and you can find those posterior distributions with INLA or MCMC. The great place to start with that is GeoBugs, or in R, you've got a number of packages. So 𝗃𝗏𝗃𝗁, INLA, which is much quicker than 𝗃𝗏𝗃𝗁. I would say that the 𝗃𝗏𝗃𝗁 is much easier to use. And the benefit of the 𝗃𝗏𝗃𝗁 package is that it's got an extensive tutorial. I've put the link in at the bottom there. So that's a really good starting place if you want to go down a Bayesian route. That's not my preference, but it's I can see the benefit, especially with complex models. It's a good, it's very efficient. So I think we're about halfway, half an hour in. I think it's probably a bit too early. Probably missed the break unless anyone's really wanting to have one. If not, I sent around some materials that were up on my GitHub. So these are the materials that we'll be using for the second half. And you can get these by cloning the repository on GitHub, and if you run setup.r, you can get all the packages that you need. So some of you might have already done that, yeah, I think if we carry on now, and if you have done that, then yeah, feel free to kind of go along, and if you've got any questions while we're doing this, please just shout out. So what we want to do with our this workflow is shown here. We want to do some pre-processing to get the variables into the correct format. We're then going to build naive models. So that's what they're commonly termed in the literature. A naive model is one that is just your typical standard regression. We then want to test for spatial autocorrelation. So the difference between last time in the way we're testing spatial autocorrelation is that we're testing in the residuals of the model rather than just a dependent variable like we did last time, and that means that we are accounting for the covariance when we're assessing spatial autocorrelation. So that's important, but we'll kind of look into that a bit more in a couple of slides' time. So if we find spatial autocorrelation is significant, is a problem, then we want to build our spatial model. If we don't find spatial autocorrelation, then it's fine to go forward with our naive model and to go straight to the results interpretation. So I've shown that using the darker purple arrow. But assuming that we do have spatial autocorrelation and we build that spatial model, we then want to compare the model in terms of its fit, and we also want to look at how it affects the coefficients, and then we want to see, interpret the results in terms of our research questions. So in terms of our research questions, we are interested in Scottish COVID-19 deaths, and we're interested in environment and socioeconomic variables. So the first question is, is there an association between long-term air pollution and risk of COVID-19 death? The second question is there an association between pre-existing inequalities and the risk of COVID-19 death? And so this, last time we set a question about how to, whether we can find evidence of COVID-19 being a syndemic. And so this question is answering that one. Sorry, this question is the same as that one. And the third is whether we see a multiplicative effect of air pollution and inequalities. So if you live in an area that has high air pollution and high pre-existing inequalities, do we see a kind of exacerbated risk of death more than the sum of the individual parts? Okay, so where does the death data come from? It comes from the National Records of Scotland. So they take the data for when COVID-19 was mentioned on the death certificate, and they provide the data for public use at an intermediate zone level. So last time we used data zones. So there's around 7,000 data zones in Scotland. So they are typically considered a neighbourhood level. Intermediate zones are the next level up. So data zones are nested within intermediate zones, and they correspond to around 4,300 people living there, and there's around 1,300 in Scotland. So on the right-hand side at the top there, you can see the deaths per week involving COVID and the week number at the bottom. So you can see that week 17 had the highest number of deaths with 661, and then thereafter it's declined with a long tail, with only 5 deaths recorded at the end of the period that they released the data. So that was the 13th of September. And so that corresponds to around 48 deaths per 100,000 on average. But you can see that the distribution of deaths for intermediate zones is very skewed. So if you look on the bottom right-hand side, you can see the graph which shows that we've got a very high number of intermediate zones that register 0 or 1 death. And so for this type of response, a negative binomial model is appropriate. And we might consider using a zero-inflated negative binomial model because we know that 19% of the intermediate zones haven't registered a death. So the zero-inflated negative binomial model splits up the regression model and fits the excess zeros separately from the rest of the data. So our air pollution data, it comes from DEFRA, which provide background air pollution data at a 1 kilometer grid level. It's long term, so it's over the course of a year. And they provide this for a number of pollutants, but we're interested in NO2. Some previous work that we've done suggested that NO2 had the most significant effect on COVID-19. So NO2 is mostly produced by fossil fuel combustion from vehicles and power plants. And there's a huge body of evidence that's shown that air pollution is causally associated with hypertension, cardiovascular disease. And why that's important for COVID-19 is that these are the same comorbidities that are associated with death. So you can see at the bottom there the hazard ratios showing the effect of having one of these comorbidities and the risk of death. So it's plausible that long-term air pollution has caused susceptibility, and that will increase our risk of COVID-19. The other question that we're interested in is whether we're seeing what's been termed as a syndemic. So on the right-hand side, you can see this is the wider determinants of health model, showing the different layers that are important for health. And really, the paper by Claire Bamborough is making the point that the same inequalities in these variables are also the cause of COVID-19 deaths. So what we can do is use a proxy for health inequalities. We can use the standardized mortality ratio. So this is a part of the Scottish Index of Multiple Depredation from 2020. So last time we used that as a outcome. So now we're using it as a covariate. And yeah, we can, if we see a relationship between ASMR and COVID-19 death, then we can say that there is the deaths are related to existing inequalities. So now on to our code. We've got some pre-processing to do. So what we first of all want to do is read in our COVID death data. You'll see that I have started working on this these materials earlier on, and so I used an earlier data set for deaths that didn't include up to September. But actually we want to use the most up-to-date data. So that's from week 17, which is up until the 13th of September. We read that in using the read.csv function. We skip the first 2 lines. There's some information that's some kind of metadata that they've included in the CSV. And we also use the square brackets to kind of slice the data, so it's just the data that we want to use. So this is actually a really important part of our programming, where we're using this to get at individual cells within a data frame. So the first number is the rows, and the second number is the columns. And the colon means that you're wanting columns from 1 to 6. But this is really kind of a useful way, especially when you are starting to write loops and also starting to define your own functions. It becomes a really important way to get data from data frames. So I definitely recommend looking at that type of indexing. I think it definitely improves your coding if you're able to do that. Next, we want to read in the spatial data. Like last time, we're using the simple features package, and we use the function read_sf. This time, unlike last time, we're using the intermediate zones rather than the data zones, and they're held as a shape file. And what we want to do then is just merge them, and we merge them on the variable interzone from the spatial data set, and we merge them on the intermediate.zone.code variable in the COVID deaths data. So it's important that if we're merging data sets that have different names that are your key, that you make sure that you do that with the 𝗵.x and 𝗵.y options. Okay, next. So we next want to do some pre-processing on our Airfusion data. So this data comes for the whole of the UK, and it comes in a point. It comes for individual points. So we need to do some processing to make that applicable to our areal data. So what we do is we read that CSV in using that first line. Again, we've got some metadata that they include in that CSV file in the first 5 lines, so we skip those. This data set defines the missing data with the string missing. So we want to tell R that that string means missing, and in R we encode missing data with NA. The third line because when the data was imported into R with this string, R thought it was a character variable. So this line here is saying that actually, now that we've assigned the right way to show missing data, it actually is a numeric data set. So it's important here just to remember that I guess the order of your code is important. So if you did the third line before the second line, you would get your variable would just be filled with missing data, and so yeah, it's important to remember just to keep in mind the order that you're doing your commands. Okay, so what we want to do next is we've got this CSV, we've got the points that relate to some spatial information, but we don't have the spatial, we haven't made it explicitly a spatial data set. And what we need to do there is use the SP package, and the components of making it spatial are the coordinates. So that's X and Y. So that's relating to the eastings and northings. So that's akin to longitude and latitude. We're not using the WGS84 projection. We're using the British National Grid projection. That's why they're eastings and northings. The second component is the data. So whatever column in your CSV that is for the data. So here we're saying that it's the fourth column is the one that has the NO2 measurements in it. And a third component is the coordinate reference system. So that's, like I said, the reason we know these are northings and eastings is because we're defining the projection. And so this is the projection string that you can get online for British National Grid. So once you've got all those together, all you need to do is run the spatial points data frame function. You supply the different parts. So there are your coordinates, there's your data, there's your coordinate reference system. And that gives you your spatial points data frame. So you've got a spatial data set now. Like last time, what we do is if we want to do further changes to an object in R, we can use the dplyr functions and code. So this is what we talked about last time. This is a pipe. So you pipe in an object to another function. So we're taking that spatial object and we're piping it into this piece of code here. I talked last time as well about there's a number of packages in R that you can use to kind of represent spatial data. So SP is one of them, it's the older package, and SF is the newer package. And for the functions in the future that we're needing to use, we need an SF object. So what we're doing here is just saying, can you convert that SP object to an SF object? And that's just a small kind of requirement for the kind of models that we'll build later on. Finally, all this is doing at the end, this is a bit overkill. But what you're doing here is just double checking that we've got the right coordinate system. Some of the kind of most infuriating issues in doing this kind of work is not having the right, having a spatial mismatch. And often that's the case. The reason behind that is because you're using data sets that have different spatial projections. So here I'm just making double sure that I've got the right one. And you notice here that I'm using a code rather than a string. Actually, these two are exactly the same. And I just wanted to show you that show that this is an EPSG code, sorry, ESPG code, which it just means that you don't, there's probably, you could have issues if you make a mistake with some of these strings. They're more prone to, having left out characters, for example. So the codes, the EPSG codes, are good to use to get around that issue. And they're all online. You can find them quite easily. Okay, spent a bit too long on that one, but onwards. The next bit of code is like one of my favorite bits of code that I use, because it's just so elegant. If you've got, so I said that the DEFRA data comes for the UK, but our study for this question is Scotland based. So you want to subset your spatial data to a smaller area, so to Scotland. And it's really eloquent because it's that one line of code there. So we can see what we're doing is it's the same kind of way that we did the indexing earlier. We take our UK-based model, UK spatial model, and then we take the boundaries that we've got for our intermediate zones, and we put it in the square brackets. So that's really saying is, what that's saying is that we only want the rows that are are in the intermediate zone boundaries. It's a very elegant code, but I can't explain it very elegantly. Anyway, so that's it happens all the time, I guess, in spatial analysis, that you want to cut your, either for the efficiency of the code, or your study is actually smaller than the area of the data. It's such a useful piece of code to know. If there's anything from the pre-processing that you use again after this, it's probably this bit of code. Okay, so now we've got, what we have on the left-hand side is the points spatial data set for Scotland. So you can see it on the left-hand side here. I've just plotted it. And you can see the NO2, which is in micrograms per meter cubed. But we need to make that, we need to assign those to the intermediate zone, because we're going to run our model using that data. So we use a package called stars. Stars is like the raster equivalent of SF. So SF deals with the vector data, stars deals with the raster data. So what you do is you take that point data and you rasterize it. So you define the boundaries of the cells, the raster cells. So that's 1,000 meters by X and Y. And then you get that, you get that raster data set, which we've termed NO2 underscore RST. And if you plot it, here's what you get on the right hand side. So now so this is an important thing to consider when trying to assign exposure, environmental exposure to boundaries. If we use the point data, we might have boundaries that don't include a point, and so we'd contribute to missing data. But if we have raster data, we can, and it's complete over the study area, then we'll we use all the information that's available. And that's what we've done for the next part. So this leverages a developer package. So unlike the other packages which you can download from CRAN from the install.packages function, this one you need to install via GitHub. So you first of all need the DevTools package, so you can download that on CRAN, and then you use the install GitHub function, and that's just all this string is his username and then the repository location. So if you know, I put a function in the spatial association repository that I linked to earlier, it would be my username 𝗵𝗿𝗃𝗃 / and then the spatial association. Yeah, so that's one way to kind of also share your functions that you build, and it's also really neat that you can link other people's functions into your work really easily. So once we've installed that, we'll load his package up, and so the package is useful because it is allowing us to have this function called Raster Extract. What that does is it says that this data set here, the raster data set, and we've got our spatial COVID death data, so that's the boundaries. So what is the air pollution in those boundaries? And we want to take the mean of the raster cells, and we want to exclude any cells that are missing. And we output that as a data frame, and then we bind that to our original data set. So now we've got the pollution, and we've bound it to our data that includes the deaths. Again, this is just a bit of code to say that we've to make sure that we're using a numeric variable, and to get this nice, I think, you know, a good suggestion for everyone that's doing this kind of work is to just constantly be plotting your new objects, and just make sure that you're getting, you know, the right, you know, what you expect. So here, what I've done is used a package called MapView. So MapView is really nice because it gives you interactive graphs and maps. Sorry, interactive maps. So you can see when you do this in R, you'll get a window that comes up, and it just allows you to zoom in. You know, so it's a bit tricky to kind of, in the last map, it's quite kind of tricky to see what's going on in Glasgow. You know, having an interactive map, you can actually zoom in, and you can then compare. Also, you know, another feature is that you can actually look at different background maps, so you can actually kind of cross-reference it with what you'd expect. So you might see that there's, you know, a factory somewhere in Glasgow, and you might think, well, you know, does that correspond to higher levels of air pollution? So you can do that when you've got that background map loaded. Okay, so we've got our air pollution, we've got our COVID deaths data. We're now interested in our measure of health inequalities, and that's the the standardized mortality ratio. And yeah, again, so because, as I said, intermediate zones are data zones are nested in intermediate zones, we need to have a lookup table so we can convert from data zone to intermediate zone. So the problem here is that the SMR data is at data zone, and we need it at intermediate zone for a model. So we use a lookup table. So the lookup table says this data zone is in this intermediate zone. So it's got a column for data zones and a column for intermediate zones. And we'll use that information to get the data intermediate zone level. You know, and then we also load in our SMR data here. We didn't talk last time about functions, but functions are really important for doing kind of repetitive cleaning, or also it has a secondary benefit to making your code look a lot cleaner. So I've just got an example here where, annoyingly, when they provide the CSV file for the SIMD, in the columns you've got what the units are. So for SIMD you've got percentage sign. So you need to take these out so that R knows that this is a numeric variable. And the way to do that is to use a package called stringer and replace all the basically take all the strings and replace all the all the special characters and replace them with space. So the only thing that I guess we haven't talked about as well as in this code is the 2 colons. So this is quite useful. The two colons are used to identify which package the function comes from. So it's quite useful if you're rereading the code to understand that it came from that package, so you can then look at the source code to see if you've got an error. The other really important point is, and this does kind of break a lot of code, is when you've got packages that use the same name for different functions, and this happens to dplyr and plyr. So and actually you do use those quite often together. So you can see here that when you want to be explicit that you're using the function from a specific package, you use the 2 colons. Okay, so this is just a kind of a pipe to show our different cleaning stages. So we kind of did this previously as well, so I don't want to spend too much time on it, but you can see there that I've applied the function that we've written up here. So I'm saying that for these 3 variables, I want to remove the special characters, and I'm removing the special characters based on what I've said to do that in this function. Okay, so basically you then you then want to. This is the important part for getting from data zone to intermediate zone. So you merge the data based on that lookup table, and yeah, again, what we want to do is get the mean of the data zones that are in the intermediate zone. So we first of all have to group the data, and then we take the mean of these variables. So I think the only kind of difference between what we talked about last time and this time is that I'm using the mutate_at rather than mutate. So that's useful if you just want to apply a certain function to specific variables rather than the whole data set. So that's worth remembering that if you append it with the @, then you can specify just individual variables. Okay, so that's nearly us done with pre-processing. All we need to do is bring them all together, and we do that by joining our COVID deaths with our COVID deaths, which includes the air pollution and the inequality data, And we're taking into account that they have different ways of showing the, different ways of representing the link, the key variable. And yeah, again, what we, like last time, there's a bit of missing data, and we want to do a case complete analysis. So we run this code to take out the missing data. And so we lose about 16 units. So 16 out of about 1,300. So yeah, I haven't looked into any more detail about the missing data, but it's so small that it's probably not too much of an issue. Okay, so that was the pre-processing. We're now on to building naive models. So this is a simple regression. We're using the PSCL package. So this allows us to do zero inflated negative binomial regression. On the left-hand side of the equation, we've got our dependent variable. So we've got our number of deaths. We've got a tilde, which kind of shows the boundary between the left-hand side and the right-hand side. And then we've got our covariates. So we've got NO2, we've got the income rate. So that might be a general measure of SES, which could be an important confounder. So that's why we've included that. And we've got total population as well, given that that could be an important confounder as well. We're specifying that the data that we're using is this object, it's contained in this object, and our distribution is negative binomial. To get a summary, to get the coefficients, we run the summary command. We also, might think about scaling the coefficients so that we can see the relative effect of each. And so that's all I've done there, is take the scaled. This code here, I also wanted to run it with a different model and also test the spatial autocorrelation on this model to see if there was any difference. So this is just a sensitivity test, which in the end showed that it didn't matter if we're using either this one or the one we've chosen. So that summary call will give you something that looks like this. You get a summary of the call at the top. You get your residuals next, and then you get your estimates. First for the negative binomial part, and then you get it for the zero inflated part. So yeah, you get your estimate. All the usual things that you get with your regression output, and your asterisks showing how statistically significant it is. So, a useful thing to do if you're running lots of models is to try and standardize the output. And this is just a bit of code that takes the the terms that we're interested in, takes the coefficients. It's just taking that column there, and it kind of brings them together in the data set. The other thing that we want to do, and this is a negative binomial model with a log link. So if we want to express the coefficients as incident rate ratio, we need to exponentiate the coefficients, and so having a function to do that, and so that you've got a nice kind of standardized output is very useful. So that's all we're doing here. We're just saying that you know, we're taking the information that you supply, and then we're formatting the coefficients so that they kind of respond, that they kind of reflect something that's a bit easier to interpret. So the IRR being a bit easier to interpret than the raw coefficient there. And then we put it in an object called naive coefficients. Okay, so now we've got the model. We want to understand whether it's, you know, if there's a problem with spatial dependency. So first of all, we get the residuals and we stick them in our original data set. We call them residuals. We then load up the SP-DEP model. This will be familiar to you from last time, where we want to understand the neighborhood correlation matrix, and so we take the we calculate that using the poly2NV. Just a word of warning, if when you're doing this, I think last time we did just Glasgow, so there was like 200, I think, areas. You know, with this one we've got 1,300, and this does take a long time. So yeah, don't worry if when you're running it, you think that things have hung up. It will run. It just takes, I think, you know, at least 10 minutes or so. So that's why I've saved it as a R data file. This is quite useful. The saving things as RDS is quite useful. It's much more efficient than saving it as, if you've got a really large data set, save it as an RDS file. It takes up about 1/10 of the memory. It loads a lot quicker. It's a really convenient, useful way to store big data. Well, not, but yeah, you know, large-ish data. So that's all I've done. So, you know, instead of running it again, I can just load up that matrix, and then we supply that to the. Before we do that, we want it in binary format. So we want to, for each unit, we want to know who's a neighbor and who's not, and we want to assign them ones and zeros rather than anything else. Crucially, from last time, which we didn't have this problem, because we're looking at a kind of area that was in the centre of Scotland. We have to say that we want to account for the fact that we have areas that have 0 links. So these are areas that are kind of like islands in themselves, so they don't share any boundaries with other intermediate zones. Okay, and then so we have to so we get that as a list object. That's what's required for our function that we ran last time as well. So that's the Miranda.mc. So it's, you know, we're taking multiple, we're running it multiple times to see how the distribution of Miranda's I plays out for 10,000 samples. And, you know, we can also plot that as well. Like last time, you can kind of scrutinize, you know, which part of the, you know, what kind of spatial autocorrelation that you've got. Is it positive? Is it negative? So you can look in a bit more detail with this Miranda plot. But if we just look at the Miranda results, we can see, you know, this is our distribution of Miranda's I for our sample that you would expect, and this is with the Miranda's I that we have. So obviously is you know, much different to what we'd expect, and that gives a very highly significant p-value. And the Moran's eye, it's not very positively spatial autocorrelated, but it's still 0.13. So if you remember from last time, autocorrelation, the Moran's eye ranges from -1 to 1. So -1 is complete negative spatial autocorrelation. +1 is complete positive spatial correlation. And if you remember the chessboard diagrams, I think they're a really nice way of kind of understanding what pattern you might be seeing in the data that you've got. So we've confirmed that, you know, it is important to consider spatial dependency. So what we need to do is build a spatial model, and it's finally time to open that can of spam. So you need to use the library command to open spam. Remember, it's 2 M's. It's not the original spam. It's the new improved spam. And with spam, it will allow you to use the fitme function. And so again, we've got a very similar setup to what we had in our naive model. So we've, you know, it's exactly the same up until here. And this is all this is doing is it's putting in the random effect. So it's putting in our intermediate zone as the random effect. And we're also including the matrix that we created. So, you know, defining which ones are neighbors, which ones aren't. And yeah, again, we just use the data and we specify its negative binomial, and we can see the results when we run the spatial, the summary command. So again, you know, if you're running lots of models, it's always good to try and standardize the output. And so, you know, we run that function that we created called output coefficients, and that gives us the model results for the spatial model in the spatial coefs object. So now we've got an object that contains all our information that we're interested in, called naive coefficients, and now we've got the spatial one as well. So it's useful, you know, just in terms of having it very standardized, but also, as you'll see, we use these objects for our plotting. So, you know, it's kind of good to just do this work previously so that you can run an easier command later on. So yeah, this is very similar to what you saw with the summary from the naive model, and yeah, you can see your estimates there. Good, yeah, and some model fit statistics at the bottom there. So now we want to see what difference it's made. So we're going to look at comparing the observed deaths to the fitted deaths, and we're going to take the fitted values from the spatial model and then the non-spatial model. And we use ggplot, grid extra. So ggplot is the kind of, yeah, like we used last time, when I said is the kind of primary plotting package in R, and the way we use it is we kind of we say what kind of visualization we want, and then we you know, we put in the data that it corresponds to. The grid extra is, just so we can see them side by side. So we're just specifying that we want 2 columns side by side with both of the graphs. So you can see here, this is the output. So on the left, this is the non-spatial model. So you've got deaths you've got observed deaths on the Y, and you've got fitted deaths on the X. On the right-hand side, you've got the same thing, but this time you've got the fitted deaths from the spatial model. So immediately you can see that there's much better model fit, in terms of being close to what we actually observe. This is quite a nice way of just seeing that better model fit. Of course, you can kind of delve into statistics for actually evidencing that as well. But this shows that the spatial model was a good idea in terms of the model being a good fit. What else we can do there is actually look at how the model. So that was an overview of the model, how it was, but we might also want to look at how it varies spatially, and so we can look back at our Glasgow. Look back at just looking at Glasgow. So we can filter our model just to see the Glasgow data. And again, we use map view so that it's interactive, and we define 3 different objects, one on the observed deaths, one on the fitted naive, and one on the fitted spatial model. And we use a package called LeafSync, which allows us to sync the maps together. So if you move one map, it just means that it's easier to move around the map and see the differences. So one thing I'd warn with doing this is that if you try to do this for the whole of Scotland, unless you've got a really impressive computer, I think it's probably going to crash. So it's much better for smaller subsets to look at this interactively. And this is what you see. If it was in the in our studio, whenever you moved your cursor onto one area in one, it would move it correspond to moving that with the other maps. If you zoom in on one, it will zoom in on all 3. So like I said, it's very useful for just pinpointing where you see the differences. So here we've got the observed, here we've got the naive. So you can see there's quite a lot of differences. So, here the naive predicted that this place here would have high deaths, where in fact that there was fairly low deaths. Sorry, I should have mentioned that the scale here is darker colors showing lower, and the lighter kind of yellowy colors showing higher deaths. But you can see, just like we saw in the 𝗔𝘀𝘀𝗋𝗋 plot graph, that the observed and spatial model correspond really well together. Okay, so we also might want to look at how does that affect our coefficients. So we know we've got a better fitting model, but does it actually matter? Like, do we see different inference? Whether we run a spatial model or a naive model. And to do this, we get our standardized data set that we've made from using our function, and we want to look at the x-axis being the model name, so the spatial or non-spatial, and then we've got our coefficient, which is the incident rate ratio. And because we're looking at three different, because we're looking at three different covariates, we want to color them differently, and that's important that we use this function here, position dodge. It means that the covariates will be separated from the graph. So I can show you what that looks like in the next one. Next slide. So that's what I mean about the position dodge. So if I didn't put that code in, you'd have the green, orange, and blue all in one group. But with position dodge, you can actually see the coefficients separately. So what we see here is that, and this is what has been said to be the case in the literature, that if you apply a spatial model to a relationship that requires it, so you've seen spatial autocorrelation, it can have effects on the coefficients, but they're not always in the same direction. So, for example, you've got NO2 that's actually become a bit stronger in the spatial model. You've got income that's actually stayed around the same, and then you've got population that's lowered in its effect size. And this is important because you can see if it doesn't make a difference for NO2. Our kind of inference is the same, that higher NO2 corresponds to higher risk, and the same with income. But you can see that our level of significance has been crossed with the total population. So you could wrongly discount total population. You could wrongly include total population in the naive model, and that would be incorrect. So that's important to remember. Okay, so just to wrap up, we have looked at 3 questions, and these are the results. So I've taken them from the scaled side to the to actually show what it is per unit increase. So you can see that air pollution is associated with 5%. So 1 microgram per meter cubed increase is associated with 5% increase in COVID mortality risk. So that's important. The SMR increase by 10% is associated with 15% increase, but there was no interaction between the two. So what can we say in relation to our research questions? There is an association between air pollution and long-term air pollution and risk of COVID-19 death. And this is really consistent with what we found in England and Wales, which we published earlier in the summer, using a different level of geography, different confounders. So that's another way in which ecological designs can be strengthened, in that you look at different populations and you kind of blind each other to the choice of confounders. It's one way to try and make the result more robust. What we found is there's a strong association between the pre-existing inequalities and risk of COVID-19, but there's no kind of interaction effect. So it's not, the effects aren't higher than the sum of the parts. They are, there's no kind of exacerbating effect there. Okay, so I just want to go through now what we've done today. We've looked at geographic correlation studies and we've positioned them within the kind of bigger group of ecological studies. We've considered a couple of examples which have really changed the course of epidemiological research, and really shows that ecological studies are at the cutting edge. But they're not without their limitations, and we need to consider how we address ecological bias and spatial dependency. Some of the best ways to do that are to look at instrumental variables, look at hybrid models, and look at spatial models. And by looking at spatial models, we've addressed some of the issue of spatial dependency in a study looking at Scottish COVID-19 death. So again, like last time, there's a lot of resources out there that are useful if you're new to R, GIS, geocomputation, data science. Really recommend, if you're not interested in R, to have a look at Geoda. It provides a really good way of doing a lot of what we've done today. So the code from last time, if you want to have a look at it, is on my GitHub, and I've also put on this time the GitHub, this time all the code and files that you'll need up there as well. So thanks for listening today, and yeah, if you've got any questions, please get in touch with me. Yeah, have a look at the GitHub, and if you want to pull the data set and make changes, that's totally fine with me. And look forward to discussing more now with you.
Mark, thank you very much for your presentation. It was an in-depth analysis of a timely topic in environmental epi, in spatial epi. And I see that you showed us a lot of material that I think can be included in a publication. Probably you are preparing to include that in a publication. Yeah. Because it really shows all the details, at least the script, all the steps you have to undertake if you want to get involved into spatial association studies. Yeah. And I think the fact that this presentation is recorded, along with the fact that all of us have the scripts and the codes for this work, it's something that our students can continue working on it even after the presentation today. Yeah. Yeah. And as you also mentioned in the past, you are also available to contact you via email if they have any question, any clarification.
Yeah, of course, definitely. Yeah, be happy to. Yeah, I think that's definitely a key part of making all the scripts and data available is that the work can be reproduced and it can be improved upon. So yeah, definitely keen to hear what everyone would like to do next with it as well.
Yes. So let's open the floor for any questions or comments. Anyone would like to ask a question or make a comment? Maybe I can ask a question in the meantime. Mark, I think that question maybe is relevant and will be useful to all, I think. Do you think that when you are considering data from different sources, where you have different levels of resolution, like many times it happens when you get data from secondary sources, are there any classification criteria, or no, better say harmonizing criteria to include in order to put together and integrate these different data sources?
Yeah, so I guess if you can do that with a multi-level model. So
let me make one more clarification. Maybe it will be easier to understand. Yeah. Let's say, for example, that you have some data that the unit of measurement is at zip code level.
Yeah.
And then you might have some other data that are at the district level or municipality level.
Yeah.
How do you treat that?
Yeah, so I think there's two strategies that I would go for, and I think you've highlighted the issue of the modifiable areal unit problem. So the MAUP is when you have different inference and it's based on the scale or the shape of the unit that your data corresponds to. So one of the things to get around that would be to repeat the analysis using data from different spatial scales, if that's possible. If it's not possible, then I guess the idea would be to increase the levels in your multi-level model so you could have one level which is the zip code. You could have the next level, which is the local authority. You could have the next level, which is the kind of region, and you can have covariates at different levels. And so that would be a concise way to bring all that together in one model. Yeah. So.
Thank you. Thank you. Comments, questions from the audience? Maybe, I think they will need some more time to look now at the presentation into the script.
Yeah.
And I'm sure they will come back to you with more questions maybe later.
Oh, yeah.
Days. Because it's already 3:44 and some of them have to go to class.
Yeah, that's okay. So, yeah, no, look forward to hearing more. And please get in touch if you've got questions afterwards. I know it's a bit difficult sometimes, just with Zoom meetings to kind of have a one-on-one. So yeah, definitely just get in touch if you want to talk about anything that I've talked about today.
Thank you. Thank you for your availability, Mark, and for your time today. I'm going to thank everybody again for their presence here. And we're going to have another webinar in the next weeks or so. Stay tuned. Thank you.
Great. All right. Cheers. Bye.